Okay, students, we can start now. There was some glitch of the signals. Good evening. Good evening. Yeah, Mehak, I had a good day today. Um, we are having external practicals nowadays. So we start with chip essay today. We are done with the protein, molecular biology, restriction enzymes. All things were done. Now today something new. So, anyone knows about chromatin immunoprecipitation technique? Anything? Anyone have you done this? This chip assay before? What? Anyone of you have done chip assay before? No answers. Okay, we will start something about it then. So basically, this is done to check the interaction of DNA and protein, how your DNA and protein are being interacting within your cell, and where they're interacting, how the interaction is done. To do so, we do this chip assay. So in the chromatin precipitation, what we do, we have cells, and the cells are cross-linked um, with the antibody binding assay. So we first lyse them. So we have breakup of DNA, and then antibodies binded to them. And after the antibodies are binded, we washing step is there, and you can see your DNA with the proteins interacted to them. They are being bounded out, and then you cross-link the reversal, then you purify them, and you have quantitative PCR, and then you have DNA and protein analysis afterwards. So not only proteins interact with one another, also the DNA interact with protein. So chromatin immunoprecipitation, it's a technique which determines whether a protein of interest interacts with a specific DNA sequence. So here sometimes proteins and proteins are interacting with each other. Sometimes DNA are interacting with protein. So all these matters. Then this technique is often used to study the sites where a particular transcription factor or histone is attached to your DNA. What is this histone modification is? Is it acetylation, phosphorylations, or methylation? So how does it work? In this chip, we can use to examine the presence of protein DNA interaction at steady state. Then we quantify that changes at the interspecific relationships with the treatment of interest. So protein and associated chromatids are temporarily cross-linked in live cells or tissues and sheared using enzymatic digestions or sonications to yield 300 to 1000 base pairs fragments of DNA. So the protein of interest along with any associated DNA fragments is immunoprecipitated from the cell's debris using specific antibody. The cross-link is then reversed and then DNA fragments are purified and the amount of eluted DNA can be assessed through quantitative real-time PCR using primers flanking the genomic locus of interest. Then DNA is amplified, then indicated enrichment in the protein of interest. And there are some other modified chip techniques. So DNA fragments purified by chip can be utilized for a number of downstream analysis techniques. Furthermore, the basic chip protocol described above can be modified to answer additional biological questions. So chip-on-chip genome-wide analysis of protein binding sites using microanalysis of purified DNA fragments. Then chip sequence genome-wide analysis of protein binding sites using deep sequencing of purified DNA fragments. Then there is a native chip in which we cross-link and use micrococal nucleus digestions to cut DNA at histone linkers to examine the DNA targets of histone modifying proteins. And also chip exo, addition of an exonucleus digestion steps to obtain increased resolutions of protein binding sites up to a single base pair. Then there's we should not go through the details. GRPET, iChip, N-chip, RIP chip. So there are various kinds of chip technologies. So go through them. It's not required. There are some limitations that a chip is have some limitations that it yields low signals as compared to the controls, giving very inconclusive data, and as is limited to the resolutions related to the size of DNA and generated low shearing, which makes it difficult to examine the exact binding site of protein. Furthermore, it's interfere with the presence of proteins at a given genomic locus. It cannot determine the functional significance of that DNA. So more, it could mask the epitome of the proteins of interest, and it's extremely dependent upon the quality and specificity of the antibody employed and may not discriminate between different DNA binding proteins isoforms. Some important factors to consider. So while the principle of chip technique is quite simple, the actual execution of chip experiment can be rather complex. Here are some important factors to consider. So garbage in and garbage out. So quality of chromatin generated in chip can make the difference between an interpretable results. So we have to take care that the limited pulse to be taken for 30 seconds at time. Antibody validation. So antibodies by the Western blots has to be recognized using denatured proteins, and chip requires recognition of these tertiary structures. Cell number consideration. Your cell number should be good in amount, otherwise you will not see the results. And finally, the cross-linking issues and appropriate control should be taken care. So let's see something about this now.
Whisper something in your ear about ChipSeek. Yeah, StatQuest. Yeah. Hello, I'm Josh Starmer and welcome to StatQuest. Today we have a gentle introduction to ChipSeek. Note, this StatQuest is based on the gentle introduction to RNA-seq. So watch that first unless you're already totally down with RNA-seq. Okay, we've got a bunch of cells. Inside each cell, there's a nucleus. And inside each nucleus, there are a bunch of chromosomes. Let's focus on the chromosomes. Specifically, let's focus on these chromosomes. Chromosomes are made out of chromatin. Chromatin is made out of DNA, plus histones, a type of protein, plus other proteins that we'll talk about some other day. They're not that important for understanding ChIP-seq. DNA wraps around the histones to package DNA, and the packaging can regulate gene transcription. Depending on how they are modified, histones can activate or repress genes. Lots more can be said about chromatin and how it's packaged, but that's for another day. Today we just need to know that chromatin is essentially DNA wrapped around histones. To simplify things, let's just use this big blue line to represent chromatin from here on out, but remembering that it's made of DNA and histone proteins. And let these big brown arrows represent genes. And let this green circle represent a histone that allows transcription. And let this red stop sign represent a histone that represses transcription. In a cell, all kinds of proteins can bind to DNA. This mustard-colored thing might promote gene transcription of this gene. And this pinkish-colored thing might repress this gene. And who knows what this green thing is doing? We can use ChIP-seq to find out. ChIP-seq stands for Chromatin Immunoprecipitation combined with high-throughput sequencing. It identifies the locations in the genome bound by proteins. That's the most important thing in this StatQuest, so I'll say it again, in bold. ChIP-seq identifies the locations in the genome bound by proteins. For example, say like we wanted to identify all the regions in the genome bound by the green thing. The first thing we do is use formaldehyde, or something like it, to glue all the proteins bound to the DNA together with the DNA. This means that all of the DNA-bound proteins, including the ones we're not interested in, are glued to the DNA. The next thing we do is cut the DNA up into small, approximately 300 base pair, fragments. Then we isolate the protein we're interested in this case, we're interested in the green thing, using an antibody. The black star represents an antibody that is attached to a bead. Then we isolate the proteins bound by the antibody and wash everything else away. Then we reverse the formaldehyde glue by warming up everything. Then we isolate the DNA by washing away the proteins, including the histones. Now that we see how to isolate DNA that is bound by a particular protein, let's take a step back. So far, the example has focused on just these chromosomes. But the process, glue proteins to DNA, cut up DNA, bind proteins of interest with antibodies, isolate antibodies, unglue and wash away proteins, applies to all the chromosomes in the cell. And it is usually applied to a pool of 6 million cells, give or take a few. So we end up with a lot of DNA fragments from a lot of cells. Then, just like with RNA-seq, we prepare a sequencing library by adding sequencing adapters to both ends of the DNA fragments. Then, just like RNA-seq, we PCR amplify the library, check the library concentration, sequence, filter out garbage reads, and then align the high-quality reads to a genome. That is to say, if this is the genome, the first read might come from here, a location on chromosome 2. The second read might come from here, a location on chromosome 1. The third read might come from here, another position on chromosome 1, etc., etc., etc. Ultimately, we get a long list of genomic coordinates for all the reads, usually between 50 and 100 million reads. And we can use those reads to create a genome browser track. These are genes and chromosome positions in the mouse genome, MM10 to be exact. This is the track that we created for our ChIP-seq reads. A lot of reads map to this region, and relatively few reads map to these other regions. This track was made from a control experiment. The control track was made by taking some of the input chromatin from the original ChIP-seq experiment, and, without using an antibody to enrich for any particular protein, ungluing all the proteins and washing them off. Then sequencing, aligning, and converting into a track. In summary, the control track uses some of the same input chromatin for the ChIP-seq experiment, but doesn't try to enrich for any particular protein binding. We use the control track to verify that a high concentration of reads in the ChIP-seq track is due to a protein binding there, and not because a lot of reads map to a repetitive region. Statistically significant peaks are usually represented on genome tracks by rectangular bars. We could then compare peaks for the same protein in different cell types, like lung versus kidney. Or, if we didn't know the specific DNA sequence that this green thing bound to, we could guess that it is a motif found in all of the peaks. Here's a motif found within the peaks that indicate where the green thing bound. The large letters are more frequently associated with the green thing than the little letters. We can also try to determine the functional role of the green thing by looking at where it binds relative to the genes. Here, we see that the green thing binds near the start of a gene, so it might regulate that gene in some way. Anyway, those are some of the things you can do with ChIP-seq. In summary, ChIP-seq identifies the locations in the genome bound by proteins. Bam! Hooray! We made it to the end of another exciting StatQuest. If you like this StatQuest and want to see more, please subscribe. And if you want to support StatQuest, please click the like button and consider buying one of my...
That was about chromatin chip. All in all, yeah, Bhavika, you are right. Where was it? So next thing, if you have time, students, please download Pymal software. So this will help you to understand 3D structure of protein. So I'm giving you mixture of biochemistry, molecular biology, genomic studies also, bioinformatics we will do also in last few days, some applications of all of them. Gel electrophorese we already did, you know. So we did something about microscope, cell culture. So all in all, within from the 1st of February we have started this, and today is 17th of February. And during these 17 days, we had touched quite a number of topics, and you have sent me really good assignments also. And I hope you also have remembered them. You know those things because today I was taking viva of the students. so they were not able to tell me that for which, like, if there is a 3D structure, you want to study about 3D structure, which kind of microscope is the best. So it's a TEM, a transmission electron microscope. What is the difference between SDS page and Agarose gel? That was very common. I mean, I want to say I should not ask so these typical questions in the Viva, because we want to just test your knowledge, because everyone has its own IQ, everyone has its own memory, everyone has its own level of understanding. And me as a teacher should not judge a student based on what they have learned, what they could memorize things. I feel pity on them because they cannot answer the things that I was asking. But on the other side, the main reason to teach you all these things was to that at the end of the day, if you are in front of any teacher or any examiner or any interviewer, you are able to speak confidently all the things that you are going through my this Edufabrica workshop, and you are able to convey the knowledge that you are getting here confidently and wisely. So please keep that mind. Don't make me feel embarrassed in front of any other student or teacher or anyone. So keep things in good hold of your studies. So now I'm gonna teach you about Pymal here. So I will just, I have very nice two videos about that. She will teach you about this software, but before that, after this workshop is over, you can download this Pymal software from pymal.org. This one. Share this software. So this software will help you to understand the structural 3D structure of protein. You can find the motifs, you can find many things, 3D structure, alpha, beta, gamma, tertiary structure, primary, secondary, tertiary, quaternary, where is the catalytic subunit, where is the active site, how, where are the alpha sheets, where are the beta sheets, parallel sheets, anti-parallel sheets. So all these things could you can learn from this. So this is the software for downloading it. And there is thing called PDB files, protein database, PDB. So this is RCSB PDB. So this is a structural view of biology here. You can see that it has resource of powered protein data bank, archive information about the 3D shapes of proteins, nucleic acids, complex assemblies. And as a member of, so you can find and annotate PDB in this. So you can, like, let's say phosphate PP1. So this was my protein PP1 I have worked for. It's very slow. This is for the protein database. You can check this later. So I will start how this Pymal software works once you have downloaded this software. So it's already in my thing also, I guess. I have downloaded that. Yeah, there, Pymal. So this is the window of that. So let's start with the video about that.
Macromolecular models are complex, and researchers have developed software that is designed for interactive visualizations of these structures. The Pymal software package is used widely to look at these structures and to analyze atomic structures of biological molecules. And it's also very useful to relatively quickly create very high quality images and even animations. So our goals for this session are to introduce the Pymal interface, load a protein structure in the software, and understand how to navigate using the mouse, the menus, and the command line. So let's get started with Pymal. On launching the application, the first thing you'll notice is the two-window interface. On Macs, the two windows are joined by default, but on Linux and Windows operating systems, they have separate windows. The top window is referred to as the external window, and the bottom window as the main window. The external window has menus across the top and a panel of buttons for frequently used functions. There is also a command line prompt for text-based commands and a message area that shows the output of commands and actions that you perform using the menus. The bottom main window displays molecules and has menus for manipulating that display. This pane also has a command line prompt at the bottom, and this is functionally redundant with the prompt in the external window. That's something that you'll find a lot in Pymal. There are often multiple ways to perform the same action. Black is the default background color, but for the purposes of this tutorial, let's set the background color to white. We can do this using the display menu in the external window, select background, and then white. To get started, let's load a protein structure. Protein structures are typically stored as atomic coordinates in a protein databank file, or PDB file. To open a PDB file you already have on hand, you can use the Open option in the File menu and select the file. You can also obtain PDB files from the Protein Databank, or PDB, a publicly accessible database for molecular structures that we will explain further in a separate video. If you are connected to the internet, Pymal can actually download and open files directly from the PDB. PDB entries are specified by a unique 4-character code. Once you know the code of the structure that you want to explore, use the fetch command with this 4-letter code at the command line prompt to load the structure. Here we will use an X-ray structure of hemoglobin with PDB code 2DN2. Now you should see the default green line representation of hemoglobin in the main window. Let's make some changes to that. Notice that after loading your structure, a new object has appeared on the right panel. This area is called the Object panel and is very useful to quickly make changes to the appearance of a molecular object in Pymal. Let's try it. We can see the gray button with the name of our object, in this case the PDB code of the hemoglobin structure we loaded. Clicking the button will toggle the display of the object on and off. Each of the squares to the right of the object button is its own menu. These are listed by the first letter of their category: Action, Show, Hide, Label, and Color. The Show and Hide menus, S and H, change the way molecules are represented. We'll switch from this lines view of hemoglobin to a representation that's better for visualizing an entire protein molecule, and a representation that may be more familiar to you. So click on the S button and we'll select Cartoon. Now we see the protein in cartoon depiction, but we still have these lines, so let's hide them using the Hide menu. Let's also hide all of the associated water atoms in the structure, which are shown in red. The L menu toggles labels. This shows chain labels. Here are the 4 polypeptide chains of hemoglobin. There are many options for labels in Pymal. Let's clear the labels to declutter our display. Next, we'll change the color from the default green color. The Color or C menu provides options to change the color of the entire object or parts of it based on certain criteria. So let's try a couple. We can change the color of the entire molecule to a blue. Or we can color by chain, which is another way, besides labels, to see that hemoglobin has 4 polypeptide chains. Note that there are a few options: recoloring all carbons, just the C-alpha carbons, or all atoms. You can explore them on your own later. Now let's talk about some basic actions in the Actions menu, which is the A square. The Actions menu is varied, but the options are sorted by similarity. Some menus are hierarchical, and you can mouse over each of these actions and get a good idea of their function. For fun here, we can compute the molecular weight of hemoglobin. You can see the value appear in the message area of the external window. Pymal allows you to interact with the structure using the mouse and was designed with the 3-button mouse in mind. So using a 3-button mouse is really the best way to have all the functionality of Pymal. But you can use a 2-button or even a single-button mouse with Pymal. You can change this with the mouse menu in the toolbar. The default mode is called 3-button viewing, and that's what we'll use here. Pymal maps multiple functions to the mouse using different modes, like clicking, holding the shift or control keys, or just holding. In the bottom right corner, Pymal shows the current mouse mode and a grid that's mapping functions to the mouse buttons. The mouse buttons are along the top of the grid, and the function or modifier or action is along the left side of the grid. The grid changes when you change the mouse mode, so you can always tell what your mouse is doing by checking this grid. To navigate the structure you're looking at, you can click and drag one of the 3 mouse buttons in the visualization window. The left button rotates the molecule. You can also rotate the molecule in the plane of the screen by clicking with the left button in the periphery of the window and then dragging along the periphery. The middle button moves the molecule relative to the center. And the right button controls the zoom. With the wheel mouse, the wheel controls the slab or the depth of field. Before going any further, let's see how we can reset the view. The top section of the action menu deals with navigation. So these options are useful in finding your way around your molecule or finding the center again if you get lost within your molecule. Here we'll use zoom, which will recenter and put the whole hemoglobin back in the field of view. The single-click options are also very useful. Let's try them. With a left click, you can select or deselect elements of the structure. Here, the selection mode is by residue. Notice how the pink squares show us that we've selected all atoms within each residue we clicked. And the messages in the external window tell us which residues we selected. On the right, you will now see a selection, SELE, with menus. So now you can apply actions, show, hide, label, and color your selection. You can also turn off your selection by clicking on its name. And you can rename it to save it using the Action menu. By clicking here on the mouse panel, you can change what type of structural element you select. Let's keep it on residues for now. By right-clicking somewhere on the structure, you activate a shortcut menu of frequently used functions that you can apply now specifically to a selected element. Just follow the hierarchy of the menus. For example, here, choosing chain, then center, centers the view on the particular hemoglobin chains on which I had right-clicked. The best ways to navigate within the structure, often the mouse is the fastest. Using the mouse motions to locate a region of interest, then middle-clicking to center it. Then drag, holding down the right mouse button to zoom in. But another way to navigate is to use the protein sequence. It's particularly useful if you know the residue number, but you don't know where it is in the structure. You can toggle the display of the protein sequence in the display menu, or you can click on the S button in the bottom right of the screen. When the sequence is on, the amino acid sequence of each protein chain within the object is concatenated at the top of the screen. Ligands and cofactors are usually at the end of each chain or at the end of the sequence altogether. Solvent molecules are also listed. This sequence allows similar mouse actions as the structure itself. So if we middle click in the sequence, that residue moves to the center of our view. Right clicks bring up the action menu, and left clicks select or deselect a residue. We can apply residue-specific changes directly from the sequence. So let's find a heme group and hemoglobin in the sequence. They are towards the end of the sequence, and the residue name for heme is HEM. To show the heme group as van der Waals spheres, I right-click, select Show, and then Spheres. Now I can see where this heme is in the structure. But if I change my mind and I want to see the bonding structure of the heme instead, I could hide the spheres, then show the sticks. Or I could do it all in one step, using the Show As menu option, which will both remove the current spheres representation and replace it with sticks. As with any software, it's important to save your work. Pymal can save a session file that includes the complete current state of your visualization, the objects, the colors, the orientation, and the representation. Go to File, then Save Session to save a session file. If you'd like to save an image, you can save a PNG or PNG file format of the current view. So we'll show you additional features in other videos, but with this brief introduction, you can now start exploring Pymal on your own. Pymal is very well documented. You can find additional tutorials, information on basic and advanced features on the Pymal wiki.
So you can do all these tricks on this software. You can learn a lot on the website also. So that was the quite basic for you, so that you can understand. This is my private chat. This is not good to see. Private life and professional life. So I share these two videos with you, so you can watch them later. They are big files. They will keep going in slowly and slowly. No worries. So my main reason for you today in this session was that you are able to understand the purification first, how we purify, quantify, how we extract these proteins. Yeah, Prashant Upadhyay, they were the proteins from Pymal only. So this made my life quite easy. So I was able to, if you go into advanced level, so you can check the distance between each molecule, each amino acids. So my work in my thesis was to identify the disulfide bridges. So I created a lot of map in that. PhD defense, like distance between each cysteine. So my task was to check with the what do you say, disulfide bridges, whether they are present or not. So what I did was I checked the distance between each disulfide bridges with the help of Pymal only. So this was mass spectrometry. So this I got these distances between each cysteine. 62. The distance between each of these two cysteines was 19.868, and 105 was this much. And distance between cysteine 105, 62 was 6 or 6.82. and we created this whole map of the cysteines. And also we created how many are being oxidized, being making disulfide bridges. So all in all was possible, and we also created some interaction between the cysteines. You can fade one part, you can make one part more darker, and create these distances. It's just about 2 or 3 days work. Once you start practicing Pymal, once you start, yeah, it is fascinating. Once you start practicing Pymal, you can generate huge amount of data in your PhD or in your master thesis, wherever you want to work with structural biology. So first you have to identify your question, what you want to look for, what is your question is, why you're doing that, and are you able to find answers that you're looking for. So it takes some time. It doesn't come single day. You need people of same liking what you're thinking. You need a lot of these work, that work is from the 3 years of PhD. It doesn't came in a single day. So it took us a lot of thinking, a lot of meetings, a lot of group discussions, a lot of expert peers discussion, and many more, many more. So that was the thing. So I will not start with the second part, because if I start with the Western blotting today, so it will take a lot of time. So today we just discussed these two parts, chip and Pymal. From tomorrow we will do the Western blotting, because if I start the Western blotting, it will take maybe 50. Do you have times of more than 10 or 15 minutes? Only then we can start. Just let me know. If we have time, then we can go ahead with it. Otherwise, you might be having classes of your biochemistry or something later after 8. So we started tomorrow then, not today then. We might not have much time, because if I teach you something very fast, it will be not understandable. We keep this limit. We keep it here today then. So any other questions you have? This we have already discussed quite number of times. It's just I want to share some videos actually, about your. Okay, before we start this tomorrow, tell me what is the southern blotting is known for? For which recognition of a molecule we do the southern blotting? It's very basic question. Yeah, great, Abhay. You have downloaded. Just have some YouTube tutorials, or go to through harvard.com, from where I've got these Pymal tutorials. You can learn a lot about Pymal within 1 week. You could be an expert actually. So many lab people, they need someone who are good in Pymal or another softwares for bioinformatical studies, molecular docking, drug discovery. So this is very necessary for the structural biologist. Yeah, right, DNA. And what is known for northern blotting? Northern blotting is for, yeah, you can start. If you have done, you can start sending RNA, right? And for protein, what we do? Western blotting. So I have this formula, S and W, S and W, then D R P. So S for southern, and for northern, W for western. then DNA, RNA, and protein. So we can recognize this S and W, DRP like this forever. So whosoever is not much aware about them, it's quite easy to understand them and good for you. S and W, DRP. Right, Western. So we will do the virtual labs tomorrow mainly. So I will just go through a little bit about their basics, not much about the presentation. I already shared away. You can check the Telegram group. It's already there. It's already there. Everything is being shared. Just check them out. That's good. That's good. So you are aware of this. I'm very happy. What? There is Eastern plotting also. This is first time I'm hearing. Is it Eastern blotting? Really? Do we have? Oh wow! Thank you. Who was it? Shashank Singh. Thank you so much for enlightening on this topic. I was just stuck with S and W so far in my life, but now I can add to my literature this Eastern blotting also for the PTMs, post translational modifications like acetylation, methylation, disulfide bridges, many more. Wow, I was not aware. So that's the beauty of teaching students, that you can also learn from them, and it's never ending process. We try to give what we have as much as possible, and we can also gain like this sometimes. Very happy to know that. I will ask the next time in the Viva and will teach them also. Eastern blotting. Okay. Great. Awesome then. Enjoy your next class. We see us tomorrow again. Same time. With these techniques, you might be knowing much of them, but still, to have a good hold them, we should go through once. Then tomorrow we will start with facts analysis, fluorescence assisted cell sorting. Yeah, fluorescence assisted cell sorting. That will be our task for tomorrow. So be ready for that. This will be a very interesting class. We will do with the two types of techniques in this. And I hope you will like the tomorrow video also with a lot of virtual videos also in it. Okay, bye bye, sayonara, tschüss, auf Wiedersehen, danke schön, thank you so much, shukriya आपका सबका.